So then I wanted to talk about the early CO metabolism and its links to the geometabolism, but we just started and analyze, and the analysis is going well, but still like maybe the conclusion will be different because it's now ongoing project. So first, I would like to talk about the unusual carbon fixation pathways in the subaqueous ecosystem. So now in the CO World project, we focused a couple different field sites. So CO2 depleted CO environment and CO2 rich CO environment. The CO metabolism is the knowledge of the CO metabolism is kind of limited because the CO is not rich in the current Earth. So then, but in a couple of different sites, like the subaqueous site, or like CO2 seep, the critical, supercritical CO2 seep. So in these kind of area, we detect CO. Not a lot, but we can detect CO. So if we can see the microorganisms in this environment, we may be able to have a hint to solve like how if the CO is the important component for energy metabolism, and we may be able to find like or like some types of trace of metabolism that we may be able to find in microbial metabolism. So, but CODH is the only enzyme that can directly react with the CO. So the CODH is definitely an important compound when we think about the CO metabolism. But the CODH, about the CODH, the next Fujishiro-san will talk about the evolutionary of the CODH, evolutionary structural evolution. So then I'm gonna talk more about the life itself, life system itself. So not enzymes, the entire metabolic system. So and the CO is a really good energy source for life. So then in chemical evolution, we sometimes don't see much about the energy or the reaction. So in the microbiologists always think about the anabolism and the catabolism. So then in the catabolism side, we always think about how the life get the energy from the environment. So then CO is a really good compound that have high energy, and the CODH can make a CO2, and the electron goes to the different way, like sulfate or hydrogen, proton or CO2, and make like H2S, H2, or acetate, or like methane. So then I've been working on the series subitization, and then because I'm gonna talk the next. So then, and life, the Earth environment is changing. That this story is also talked about somebody. So I'm gonna skip details. But before the Great Oxidation event, the Earth should be more radioactive environment. But also maybe have some discussion. But then the life on the Earth right now, most of the life is supported by the solar energy, and the very, very limited life is supported by geochemical energy. So, and then it's really difficult to know, to imagine like what is going on before the photosynthesis, because the life on the Earth right now is really supported by the solar energy. So then we look like for example, that in deep, like subsurface or deep sea, for example, deep sea, there is no light, but still like a lot of oxygen there and also organic carbon there. So those are the products of photosynthesis. Then how we can understand the life very isolated from the photosynthesis? Then I've been working on the life in a deep and then in a rock powered life. So that is kind of isolated from the surface environment. So the subionization reaction have already explained by Martina. So, but the water rock reactions, the rock is called the peridotite, and then it is a component of the mantle rock, and then the mantle rock and the react with the water, but the rock contains iron, so then an iron water reaction occur, and then the hydrogen is produced, and then also like Then when CO2 is there, the methane is also produced. So these two compounds could be the energy for life, and it could be also support the early life as well. So then I've been working on the psilocybinization for like 10 years or so. And then here is the geochemistry of the psilocybinization site. The pH is really high, like 11 and 12, and the EH is really low, like minus 750 millivolt or low, and rich in hydrogen and methane, and relatively rich in calcium ion, and poor in dissolved in organic carbons, and poor in electron acceptors. So here has a lot of energy, but it's really difficult to make a like gradient, the redox gradient, because limited the electron acceptors. So the subordination could be can occur in the Earth, but at the same time, it could be occur in the solar system as well. So the JAXA brought back the small rock from Ryugu or Itokawa, but the most main component is the olivine. So, and also Mars or like probably like Enceladus. So that in those kind of stuff, in those kind of anyway, like in a solar system, the olivine this rock is really common mineral. So the subionization could occur on the Earth, but at the same time, the different solar system as well. So then this is like a pH diagram. So the Enceladus water is kind of similar to the series and the velocity. So those kind of site could be like potential analog of Enceladus in terms of the redox and the pH. So the series, I've been working the series subordination because it's kind of like a unique in the subordination site among the subordination sites. So here is the water comes down from the top, and then in that process, a degradation of organic carbon from photosynthesis occurs, and also the reduction of oxidative compound is also occurred. So then we can get the really pristine samples and then from the deep. So subordination site often mixture like interface community and the really deep pristine community. But in this system, we could get like a really pristine community and a deep. So that is really advantage for this kind of studies. So then here it could be the earlier analog on current as. So the chemical physical constraints and driving force and biological function could be similar to those present in the earlier setting. So the biological metabolism adapted and optimized to the analog areas could reflect the biological metabolism that likely occur in the earliest. That is also like controversial with kind of the backgas like evolution, but anyway, but that is the motivation that I started the work on the subordination ecosystem. But when we think about the current knowledge, the system is really challenging for life. So for the breathing and for example, so the phosphorus and calcium is really depleted because it's calcium rich, and so the phosphorus and carbon source is really difficult to get. And also electron acceptor like oxidative compound is limited, and also proton motive force is really difficult to use because the pH is really high, so it cannot make a very good gradient. So in these reasons, so the life could be challenged to survive in this system. Then actually the cell density is also really low, like 7 times lower than the oceanic water. It's really, really low concentration of cell can survive in this system. So when we look at the genomes from the deep groundwater, we see like the 80% is CPR, but I won't talk about this. But today I want to talk about the organisms in the acetogens. So mostly like a CPR, but the independent organisms are mostly like acetogens. So 80% of CPR and 20% of acetogens, something like that. So the main energy metabolism is the acetogenesis. So here, the methanogenesis, acetogenesis use like reductive acetyl CoA pathway, which is like often discussed in this meeting as well. So then that could be like one of the proposed as an ancient energy metabolism for life. So here is the gas composition of the seizures hybridization site. But when the acetogen use basically hydrogen and CO2 to make acetate. But hydrogen CO2 can make methane as well. So why this system doesn't have any methanogens? So very limited methanogen and mostly acetogen. So when we think about the thermodynamics calculations, so acetogenesis and methanogenesis is here. So that when at the high pH environment, acetogenesis should win in this setting. No, methanogenesis should win in this setting. So why then acetogenesis cannot beat methanogen, but the reality is that a lot of the acetogenesis are there, but the methanogenesis are very limited. So I want to talk the two different organisms today. I want to introduce here. So an energy metabolism of these two organisms. So here is like a Corticobacteria called NPL UPA2, which is all really distributed in a different subordination setting. So here has an energy potential in carbon fixations. So the acetyl-CoA pathway, it has acetyl-CoA pathway. But very interestingly, the organism lack the hydrogen. So as I mentioned, like hydrogen is rich and it's saturated the hydrogen, but the organism lacks hydrogen. So it's incapable of using hydrogen. And what could be a reducing power for this organism? And when you look at transporters, we didn't detect any no organic or inorganic carbon transporters. So the permeable molecule could be a substrate for this organism. So in this case, so probably from the genetic constitution, hydrogen and carbon dioxide are not the substrate for UPA2 at the seeds. And then probably, in this case, probably like a CO could be the energy metabolism for life, because CO could be the energy compound and also could be a carbon source, and also can generate in this Fischer-Tropsch types process is intermediate compounds. So formate or CO could be the energy and carbon source, like also the metabolism. So if this organism can use the CO, that is really good energy source for life. So CO is really good. So CO is a good energy source for life, and compared to the hydrogen as well. So it's better than hydrogen. So this is really good driving force to this energy metabolism system. So if this organism can use CO, then at the high pH, so this is the energetically really favorable situation. So the concentration of CO in the deep subsurface of seabed is so far unknown, but however, the drill site, like a chromo site, contain our Oman site, so they contain carbon monoxide in the deep, about the 0.1 to 0.2 micromolar. So that would be a good energy and carbon source for life. So the next one is methanol salcinolis, called the MET12. So this is also that came from the series, and then this seems like a methanogen, because methanol salcinol is a really well-known a methanogen group in terms of the taxonomy, but the methanol salcinolis in this system lacks a lot of genes like MCR and MTR and coenzyme M and coenzyme S. So, and it lacks a lot of genes and hydrogenase as well. So this was probably a methanogen, but not methanogen anymore. And then this is the first evidence of incapable methanogenesis within the traditional methanogenic orders. So that is kind of very interesting to me, so this MET12 cannot make methane. So when we look deeper into the energy metabolism, so this organism lacks like electron bifurcation enzymes as well. So HDR related enzymes here. So the entire HDR related enzyme also removed, locked, or streamlined or whatever. So that is quite interesting. So that nothing cannot make a methane, also cannot use the electron bifurcation. So this is electron bifurcation. So I think like a Joseph in a Joseph's group, the paper say like the modern microbes that use the acetyl-CoA phosphate typically fix CO2 with electrons from H2, which require complex flavin-based electron bifurcation. This presents a paradox. How could primitive metabolism system have fixed the CO2 before the original protein? So this he also mentioned like a CO2, but if CO, if the organism can use the CO, it's not necessary to have the electron bifurcation. But if we think like the CO2, then it should have the electron bifurcation. So I thought first like that this organism use CO again, and not this H, not it should use the CO. Then it's much easier to make this process. But that wasn't, because I found like the high affinity bicarbonate transporter in the genome. So it's wanted to have the CO2 from the environment. Then this, and then actually the carbonization site has a very similar CO2 high affinity bicarbonate transporters. So many of the carbonized organisms have the similar bicarbonate transporter in the carbonization system. So it's the one of the organisms is called the serpentinimonas, which is like a at the interface area. So oxic interface. So it was easy to grow in a culture. But the enzyme in our transport is really distributed in the, between an anaerobic and aerobic. But anyway, the many organisms have this electron bicarbonate transport. So here is this serpentinimonas that was isolated from this area, and then this organism can use the calcite as a calcium carbon source to get the carbon from the outside. So this is not necessary. So then when we look at the water, so we didn't detect any like a bicarbonate in the system, but this organism can get the carbon from the calcium carbonate. So that could calcium carbonate the carbon source, and then hydrogenate the energy source. So this organism really fit to the serpentinized area. And then this organism has the SBT, the high affinity bicarbonate transport, similar high affinity bicarbonate transporters. So the MET12 should use the CO2 for the carbon fixation with that electron bifurcation. That was kind of the mystery. So when we look at the metatranscriptomic data, we found that the two genes are highly expressed. And then, pillin and MMCX. So the MMCX is a multi-heme C-type cytochrome. So, and the pillin, so that's the microbiologist who knows about the electron microbiology. So think about, oh, this organism might be able to use the electron from the outside. So then I wanted to prove that. So then I used the Schwanella ordinance MR1 to determine the function of this, the cytochrome, multi-heme cytochromes. So time is coming. Anyway, so then this is the Schwanella system. So the Schwanella has an MTR, OMCA, and as the main component to get the electron from the outside and also throw out. So donating reaction and accepting reaction, both reaction can use this MTR and MTRC and OMCA. So this organism use the electron as electron donor, and also this can use the electron as can surround the electron accepted. So then we disrupt these genes and then replace to the MMCX. Then when we look at the electrochemical, when we did the electrochemical analysis, then we found that the MMC, if we liberate the MMCX, that can really the current density is really getting much higher than the wild type at minus 400 millivolt versus standard hydrogen electrode. But so coupled with the fumarate rate reduction. But if this case is the electron is an electron donor. So in this case, electron is the electron acceptor. So in that case, this MMCX cannot donate the electron to the outside, like to do the lactate oxidation. So at the lower potential, it can work, but the higher potential, it cannot work. So I mean, like MCX has an electron accepting capacity, but not donating. So then this enzyme has a similarity to the couple of different like nanowire like structures. So this extracellular cytochrome nanowires appear to be ubiquitous in a prokaryote. That is published in the Cell in 2013. And then when we did the AlphaFold analysis, the MMX also have similar. And the alignment, the sequence alignment is also similar, and also to the nanowires. And then structure is also kind of similar. So the MMCX is like get an electron and then use to get the electron from the outside. So that could make this system work. So then this travel-like organisms are really distributed in a sub-Bethenite system. So this is kind of a common system. And here is the electron-fueled acetogenesis, they call like sub-Bethenite of the meth trail. But the potential electron donor is like some mineral, like highly reduced mineral, probably the foreign site, but maybe I don't know. I want to know the mineral, just like what is a good target for the mineral outside. And then potential carbon source could do bicarbonate. So without electron bifurcation, this one we can do use the electron to fix the CO2. So yeah, okay. I will finish. I have covered side, but yeah, here is the acknowledgements. Ken Nelson, and then High Schoonen, and Fumio Inagaki, and the Shunichi, and the funding sources. Thank you very much.